everyone, so in this PowerPoint I'll be going through the review questions and the suggested answers. Okay, the first question is, what is Mendel's second law and what does it state? So this is a two-part question. Okay, so the second law of inheritance is known as the law of independent assortment. So what that says is that the alleles of a gene for one trait sort independently of alleles of another gene that control a different trait. So where we really see this is during meiosis 1. So remember that during meiosis 1, well, particularly during metaphase of meiosis 1, so metaphase 1, the homologous chromosomes line up on the metaphase plate. Now the arrangement or the configuration that happens is random. So if we think of the red and the blue, maternal, paternal, how they line up is going to be quite random. So, the paternal chromosomes might line up on the same side of the metaphase plate, or, they might line up on opposite sides of the metaphase plate. So it's going to be random, and it's going to affect, obviously, the gametes that are resulting from meiosis. So this is another diagram showing that same principle. So on the other slide, we just saw 2 pairs of chromosomes. Here we're seeing 3 different pairs of chromosomes and how the configuration of these 3 different pairs could be different. Imagine when we're thinking about 23 pairs. So remembering that our cells have 23 pairs of chromosomes and they're going to have to line up. So there is such a huge amount of possibilities of how they line up on the metaphase plate. So the principle of independent assortment is that alleles are on different chromosomes assort independently. So how one lines up on the metaphase plate is very different and random and not reliant or affected by the alignment of other chromosomes on the metaphase plate. So they're very random. It's very independent of each other. So therefore, this is what contributes largely to the variation we see in the gametes that, are produced. Okay, for this question, complete the sentences below about the principle of linkage. So for this question, I just want you to read it through and see what terms you think are missing. It might be worth just, going back through the principle of linkage yourself so you get a good picture before you attempt this question. Okay, the following statement relates to linkage. So the tendency of 2 or more genes located on the same chromosome is that they are inherited together. The closer the genes are to each other, the more likely they are to be inherited together and appear to be linked. Okay, in roundabout terms, another way of saying that is if 2 genes are located on the same chromosome, if they're close together, it's likely that they're going to be inherited together with each other. Okay, if those 2 genes are inherited together, we say that they're linked. Okay. Okay, question 3. What are the possible outcomes of meiosis in a heterozygote individual with the genotype capital A, lowercase a, capital B, lowercase b? So list all the gametes that this individual could produce. So I'm remembering that I've got 4 alleles for 2 different genes. So this is telling me 2 genes, it's a dihybrid cross. So thinking about how many different combinations could that have? Okay, so the possible outcomes of meiosis, so the gametes that'll be resulting from this heterozygote individual with the genotype capital A, lowercase a, capital B, lowercase b, are capital A, capital A, lowercase a, uppercase b, and lowercase a, lowercase b. So if we want to kind of visualize that, so if I write the genotype here of the heterozygote for the 2 genes, remembering independent assortment, those alleles are sorted independently of each other, and remembering that gametes are haploid, so I get 1 copy of each gene. Okay, so if I go, let's say I've got this one and this one here, that gives me this possible gamete. All right, if the resulting is this, I would get this gamete. Another possibility is I could get these 2 come together, And lastly, I could get this allele and this allele come together, which would give me this result. Okay, so so you have 4 possible combinations that result from meiosis of this individual. Okay, question 4. For each of the following statements, state whether it is true or false. Okay, so the first statement is, genes are linked when the percentage of recombinant gametes falls below 50%. B, recombinant gametes are observed in the offspring. And C, if A and B loci are very close together, the probability of a random crossover event is very low. Okay, so that first statement, genes are linked when the percentage of recombinant gametes falls below 50%. So this is true. So remembering that if I'm looking at that A and B locus, the recombinant gametes are going to have a combination, a unique combination of alleles that's unique to them and different to the parental generation. So there needs, in order for these alleles to come together, there needs to be that independent assortment. Okay, if genes are linked, there's none of that independent assortment. Those genes are being inherited together. So what I would see a lot of in gene linkage is I'd see a lot of this parental type of gametes, but very few of these recombinant gametes. So if I have less than 50% of these recombinant gametes, that suggests that the genes are linked. So this is a key indicator. The second statement, so recombinant gametes are observed in the offspring. So this statement is, if genes are linked, I'm going to see it's very rare that I'm going to see these recombinant gametes. Okay, so that's again another statement regarding linkage. If the A and B loci are very close together, the probability of a random crossover event is very low. So what that is, again, if I'm looking at a set of chromosomes, okay, so if I'm looking at those two, if the genes of interest are very close together, okay, it's very unlikely that crossing over is going to occur between these loci, okay? The further apart the two genes are, the more likely crossing over is going to occur between them. Okay, so this slide refers to review question 5A and B. Sheep blowfly chromosome 5 carries genes for resistance to insecticide dieldrin, gene D. The same chromosome carries a gene called furrowed eyes, F. Complete the allele symbols for a fly that is heterozygous at both loci. If no crossing over occurs in meiosis, the gametes will carry either E, F, or lowercase d, lowercase f alleles. What combination of alleles will be present in gametes if crossing over does occur? Okay, so the answer here, so complete the alleles for heterozygous. So if I'm heterozygous, remembering I've got one copy of the dominant allele, one copy of the lowercase allele. So what that's going to look like is I'm going to have a lowercase d on this loci and a lowercase f on this loci. So if thinking about the combinations of alleles, so if there's crossing over, there's a couple different things. The person could have this combination, they could have this combination, this one, or this combination of alleles. Okay, question 5C. Construct a Punnett square for a cross between the fly shown above, genotype capital D, lowercase d, capital F, lowercase f, after recombination has occurred in meiosis, and a homozygous recessive fly, so with lowercase d, lowercase f, lowercase f. Okay, so what would that look like as a Punnett square? So you might be confused because this is a very small Punnett square, but I'll explain why. So first I have to remember what are the possible gametes. So let's look at this first one, the heterozygote. There's actually 4 possible gametes, remembering with recombination that would occur from this heterozygote. So the first thing is there could be one gamete that carries both dominant alleles. The next gamete could carry a dominant D allele and a recessive F allele. Then I could have the recessive allele for the D gene and the dominant allele for the F gene. And then I could have the recessive allele for both of these. Okay, so there are the 4 possible gametes. Now for this individual, Notice that it's homozygous recessive. There's actually only one type of gamete that this individual will produce, and it will be that one. So just a copy of the recessive D gene and the recessive F gene. So I'm going to put that in here. That's why I only have one row. There's no point in me doing 4 rows of the same thing. One type of gamete, so I'm just going to do it with 1 row. And that's a lot easier when I'm doing my genotype ratio. So I'm going to put it here. So my dominant allele always goes first. Then I'm looking at this one. Okay, so now I've got, it's out of 4 possibilities. So now I look at what my possible ratios are. Okay, so having a look, you can come up with a bit genotype ratio and possibly a phenotypic ratio as well. Okay, question 5D. If dieldrin resistance is the dominant trait and furrowed eye is recessive, what proportion of the offspring with normal wild-type eyes are resistant to the chemical dieldrin? So let's go through and really look at what those alleles will be and what the phenotype resulting from the alleles will be. So dieldrin resistance is dominant. So remembering that dieldrin is used with that D allele. So if this got the capital D allele, that means that the individual is resistant to dieldrin. Okay, so then that means if they have the recessive allele, now remembering recessive, they'd have to be homozygous recessive. That would be, no resistance for this particular one. Okay, then we look at the F allele. We have capital F and we've got lowercase f. Now furrowed eyes is recessive, so that means that the lowercase f means furrowed eyes. And that would mean that the capital F means normal eyes. Okay, so looking at that information, thinking about when you look back at that Punnett square, working out the phenotypic ratio. Okay, so the answer to question 5D. So again, so if dieldrin resistance is dominant trait and furrowed eye is recessive, what proportion of the offspring with normal wild-type eyes are resistant to the chemical dieldrin? So when I see that term proportion, that is telling me I need to work out the probability. Okay, so a percentage would be perfect here. So I'm going to go back through and do my Punnett square, just so you're really familiar with that independent assortment of alleles. So there's 4 possible gametes for the heterozygote. For the other individual, there's only 1 possible gamete. Okay, this is the allele combination. Fill in my Punnett square. Okay, and now I need to look at my possible results. Okay, so I'm just going to say for my genotypic ratio, I have 1 of this, 1 of this one, and 1. So this is, I've got 25% chance of each. But this question's asked me particularly about a specific trait. So what proportion of the offspring with normal eyes? So I'm only looking at those with normal eyes. So remembering that normal eyes is dominant to furrowed eyes. So I'm looking for a capital F. So I'm looking at this one over here. I've got normal eyes. And this one here, I've got normal eyes. Because of that, dominant capital F. Okay. So what proportion out of these 2 are going to be resistant to the chemical dieldrin? Now I can see that this one has the capital dominant allele for this specific trait. So this one is dieldrin resistant. If I look at the other one, that one is homozygous recessive for dieldrin resistance. So this would mean no resistance. So if I'm looking at this, I'm saying, okay, 1, I've got 1, which is normal I, and de-Aldrin resistant. And I have 1 normal I. Sorry for getting squished here. No resistance. So what proportion is it? The answer is 50%. Okay, so 50% of the offspring with normal eyes will have that resistance as well. Okay, so those are the answers for the chapter 11.3 review questions. If you have any further questions, or there are some of the questions you still don't quite understand, please email me. I'd be happy to go through it with you again and find some more worked examples to post up as well. So please get in touch. Okay, thank you.